All right, guys, we're going to go ahead and get started in a couple of minutes here. A couple of things. Dr. McDowell had contacted us. He was running a minute late. He will probably come in during we're going to go ahead and get started with our first presentation. He'll probably come in during that presentation, so we'll just kind of usher him quietly in if that takes place. A couple of things to know before we get started. First of all, just in general, give yourselves a big hand because you're done. We say this about this experiment. We have been doing this for over 1,000 students that participated in this experiment through Blue Ridge Virtual Governor School. And while you're in it, it's demanding, it's technical, it's frustrating at times for some of you, both in terms of the actual process of the designing the experiment, collecting the data, figuring out how to present that data. All of that stuff is meticulous and demanding, as is working with your teammates at times. We know that, right? Okay, we understand that. But let me tell you this. If you can do those things, if you, as an individual, can participate effectively in a process where you do something like that, design and execute an experiment, collect and present data, work well with teammates, even in frustrating situations, and accomplish something, if you can do that, you can be successful in anything you do, okay? And we have heard a lot from many of our students, many of who've gone into the sciences after they've graduated from this program and gone into college, that this was one of the first times that they participated in a real experiment. A lot of the experiments you've participated in are things that take place in the context of 1 to 2 days in the classroom. Real experimentation doesn't take place in a day. It takes place over months and years and decades sometimes. And so you guys have participated in a process like that. It's really important. So while it may be frustrating at times, it is really good skill preparation for the things that you're going to encounter later on, whether you go in sciences or not, because it's the same sort of things that we have to do in other disciplines. So nice job with that. So today during the presentations, we've got a lot of groups. We're going to have to move fairly quickly on this, so there's not going to be a lot of Q&A time. If you have questions for any poster time that we have left at the end of the day, that's the time to address those questions with the groups. Teachers may have, and Dr. McDowell may have 1 or 2 questions for each group, but we're going to have to keep things kind of rolling. One thing about a couple of pieces of just protocol. If you, during the course of presentations, whatever, need to use the restroom, quietly just get up and go. You don't need to ask for permission. You know where they are. Cell phones need to be off and out of sight. So right now, if your cell phone is on, as mine is, we're going to turn it on and turn it off and put it away. I have been known to leave at the end of the day with about 2 or 3 student cell phones. I do not want to do that today. Okay. A couple other things. These will be filmed. It'll take us a couple of days to kind of break the film out and by classes and stuff like that, but you will have an opportunity for your parents. Here's Dr. McDowell just in time. Big hand. And again, one of the things you're going to be listening, you're going to be hearing about the other schools' results. We were hopefully have a little bit more poster time at the end, so if you think of any more questions, particularly those of you who are on the poster question cruise, that's time to do that. So at this point, I'm going to turn it over to our MC for the day, Mr. Budichevsky, or Mr. B, as we call him.
I did have something I had to say, but I forgot what it was. So, again, just remember, if you have questions for the people presenting, that's something to save for when presentations are done. A lot of times when people do have questions, it's something like really nitpicky about things. Like, why did you call it rosette diameter instead of rosette width, and like stuff like that. So, or like just stuff like that. So if you do have questions, and this is not just people for the question group, although if you didn't get an answer when you went around to the posters, you may get one now. But if you have questions, save those, write them down. Something to pay attention to is, the mutation, the plant. What you should be looking for is what was the treatment and how did those results differ from the ones that you guys got in your experiment, because that's the big difference. So pay attention to that. See if you notice similarities. See if you notice huge differences. I mean, you guys have spent over a month with these plants. You know what's up at this point. So green folks, if you want to come up here, if you're ready to go and get started.
presentation team, I'm Jillian, and to my left is Stacy, Olivia, Molly, Julia, and Carter. We conducted an experiment in which we tested the effects of nickel sulfate on wild-type and mutant Arabidopsis presentations. The ACT2 gene, a gene essential for the transport of protein throughout the plant, was removed, making it illegal. We are here to present to you our results. In addition to the gene, we chose nickel sulfate as our variable. Nickel is a heavy metal that is necessary for plant growth. Although if too much is added, it could become detrimental to plants. Some of the effects include reduced reduced shoot, plant growth, and inhibited photosynthesis and transpiration. Sulfate can produce a pungent odor and strong taste. It is one of the 3 secondary nutrients required by plants to maintain normal and healthy growth. It aids in the production of proteins, resistance of disease, and seed formation. We chose nickel sulfate as our variable because we believe that it would result in documentable stressor and we made it by taking 526 milligrams of nickel sulfate and adding it into a beaker that contained 250 milliliters of water. We stirred the solution until the crystals of nickel sulfate were dissolved. The toxin was administered on Mondays and Thursdays up until there was an experimental error that caused part of the class to start administering the toxin on Tuesdays and Fridays. Each time the toxin was administered, it was administered with 1.25 milliliters of nickel sulfate, which is approximately 25 drops onto the soil. The purpose of this experiment was to measure the effect of nickel sulfate on seed production of Arabidopsis wild-type and mutant plants. We used 4 trial groups to gather more precise and accurate data. We believed that the nickel sulfate along with the gene would result in a lower reproductive.
For our experiment, we created 3 hypotheses predicting what we thought would happen based on the different variables. Our first hypothesis looked at the effects of the AAP2 gene on Arabidopsis plants by comparing the wild-type control to the mutant control plants. We predicted that when the gene is present in the wild-type plants, the wild-type control will experience shorter plant height, smaller rosette diameter, and a presence of yellowing in the leaves. Our second hypothesis looked at the effect of the nickel sulfate on the plants by comparing the wild-type control to the mutant experimental plants. We predicted that when nickel sulfate is added to the wild-type experimental plants, there will be a presence of anthocyanin in the leaves, shorter plant height, and reproductive yield will decrease in the number of seeds produced. Our third hypothesis compared the wild-type control to the mutant experimental plants and looked at the effect of both the AAP2 gene and the nickel sulfate on the plants. We predicted that if the gene is removed and nickel sulfate is added, then the mutant experimental plant will be less successful as indicated by shorter plant height, less seed production, and a presence of anthocyanin in the leaves.
In the beginning, plant count decreased overall due to plant thinning is the process of plant removal of porcelain. We chose to thin the plants to prevent overcrowding and future complications with measuring. The increase on October 4th is due to further plant growth. Leaf count decreased overall due to previous thinning. Any increase in leaf count during this period of time was due to further thinning.
Plant height. From October 11th to October 19th, plant height increased overall. And as you will notice, there is a significant increase in the mutant control. This is because when there were more plants per pot, plants weren't able to grow as tall. But when there were less plants per pot, they had more room to grow, so they grew much taller. And that's why we believe mutant control grew taller.
Rosette diameter was measured from October 17th to October 24th. This was done by using an electronic caliper to measure the distance from the outside of one leaf to the outside of the leaf directly across from it. The rosette diameter increased gradually over time. It can be noted that the wild-type plants had a slightly larger rosette diameter than the mutant plants. The mutant control plants had a considerably smaller rosette diameter compared to the other plants.
Bolt height was measured when bolts first started appearing on October 20th. The order in which the plants bolted were very important for the results of the experiment. Those that were unhealthy bolted first, and those that were healthier bolted after. This also affected the seed production later in the experiment.
The number of seed pods on each of the plants was counted starting on October 27th and continued until the end of the experiment. The seed pods were counted in order to determine the reproductive abilities of the plants. During our experiment, we discovered that a higher seed pod count indicates an unhealthy plant and that a lower seed pod count indicates a healthier plant. This is due to the fact that when plants are unhealthy and dying, they produce seeds at a faster rate and release them quickly so that they are still able to reproduce before they die. As you can see, the mutant experimental plant has a significantly higher seed pod count compared to the rest of the plants, indicating that it is unhealthy and sicker than the rest of them.
There were many types of stress throughout this experiment. This is indicated by the presence of anthocyanin in the leaves. Those that had more red in color in their leaves were more stressed than the other plants. It is also seen through the lack of trichomes, yellowing in the leaves, and the basic unhealthy signs of plants, which include brittleness and wilting of the leaves.
There were three main sources of error found in our experiment. They included overwatering, photooxidation, and human error. The soil developed a white fungus due to overwatering. This fungus competed with the Arabidopsis plants for nutrients. Photooxidation occurred due to the 24-hour light given to the plants. The excess UV radiation caused a reddish color in the plant's leaves. Smaller leaves that were covered up by larger ones were found to be still green, as indicated by this arrow. This is because they were protected from the harmful UV rays. The human error found in our experiment as well. In the beginning, there was miscommunication on how to measure the plants. Some groups used rulers, which were less precise. Finally, it was agreed that electronic calipers should be used for more accurate measurement. There was also miscommunication in regards to leaf diameter and rosette diameter. Some groups measured leaf diameter first and then rosette diameter when there were more visible rosettes. However, other groups measured rosette diameter from the very beginning. This caused data to be inconsistent between the groups.
The design change. What we would recommend for a design change would be trying to limit the amount of outside variables like the overwatering and the light. To do so, we would recommend giving them 16 to 18 hours of light as opposed to the full 24. And for overwatering, we would recommend testing the soil to see whether it's damp or not instead of just looking and assuming. Another thing we could do with the design change was after our Skype conversation with Professor McDowell, he posed another hypothesis about bolt time being directly related to the seed pod count. If we were to change that to our hypothesis, then we could use this as a future application and run another experiment. The importance of this experiment. The reason this experiment is important is because we had a lot of differentiation amongst our groups and even inside each group. And again, after our conversation with Professor McDowell, we were kind of wondering why this happened. So he made that hypothesis and said potentially Virginia Tech could maybe go back to the drawing board, and they might be wondering why what happened did happen. Another reason this is important is because farmers could use this. They could be able to look at whether the gene hurts or helps the plant more, and maybe being able to resist a certain disease and going with whether that gene is removed or not and planting that crop.
In the end, our first hypothesis was rejected because while there was a shorter plant height and a smaller rosette diameter, there was no significant yellowing in the wild-type control plant when compared to the mutant control plant. Our second hypothesis was also rejected because while there was a presence of anthocyanin and a smaller seed pod count, the wild-type experimental plant was actually taller than the wild-type control plant. And finally, our last hypothesis was also rejected because while there was a smaller plant height and a presence of anthocyanin, mutant experimental plant had a higher seed pod count than the wild type control plant.
So some final acknowledgements. We would especially like to thank Professor McDowell for all the time that he spent Skyping us and answering a lot of questions and giving us a great opportunity to be able to participate in a lab at the collegiate level. We'd like to thank Mr. Caraway and Mrs. Oughton for setting this up and making it happen, and thanking our teacher, Mrs. Dexter, for helping this experiment go really smoothly inside the classroom.
Teacher, Dr. McDowell, any questions?
So if you had one thing to do over again, what would it be?
Probably the light and watering, because I think potentially that could help change doing 16 to 18 and watering them when they actually need to be.
Great. I really appreciate the critical analysis that you subjected your work to. That's good science.
That fungus that you said grew on some of the plants, did that grow on all of them or just some of them?
They grew on all of them. It was in the soil. It didn't really have to do with the plants themselves.
We're going to have to move it on just because presentations, good questions, good presentation. Thank you. Thank you.
Thank you.
Next up, I believe is Goochland 3. Keeper of the microphone. Right. Mr. B, you got him? All right, ladies and gentlemen.
Hi, my name is Firstly, and this is Garrett, and this is Keyshawn, and we did our experiment on Arabidopsis thaliana and salt pollution effect.
So we use Arabidopsis thaliana because it's a model plant, it's a short gestation period, but we also use it because it has a very short genetic sequence. And researchers have found this gene called AAP2, the AAP2 gene, which inhibits a pathogen named HPA from infecting the Arabidopsis plant. And we wanted to find out, what else does this AAP2 gene affect and see what other benefits it might hold or other detriments.
Our hypothesis was if you add 4 grams of salt for every liter of water, it will then decrease, the GMO will then do worse in the salt water than the wild type. We believe this because of the inhibited proteins in the cell membrane would then cause less water to be able to re-enter the cell after the osmotic stress has pulled it out. The variables we chose salt water because it has to do with that movement through the cell membrane, and it also is a very important thing. There's lots of areas where it's very prominent. We put them both under the same lamp for 24 hours a day. At the very first 3 weeks, we both gave them fresh water, 300 milliliters. And after the first 3 weeks, we started adding salt water, which then we increased both plants got 400 milliliters of water, but we noticed some signs of stress on the plants, thinking they might be dehydrated. This is the rosette size. As you can see, the different things, the wild type control, it's just the wild type, the control is the fresh water. Rosettes are 2 leaves that are facing opposite directions, and you measure the distance between the 2 tips of them, and then we average them along the class, and you can see the results of our data.
In this, it shows the number of bolting, and bolts are stems that came from the plant showing the reproductive stress. And as you can see, the wild type experimental had the most number of bolting, and the bolting occurred later in the experiment.
So So by this graph, you can see this is reproductive yield. And we measure that by seeing the amount of soliloquies on each pod and then averaging it. And as you can see, the mutant control had the most number of soliloquies, which results in an increased reproductive yield, while the experimental mutant has less, but that's mostly just because it was placed in salt water, which is naturally a stress eater, and it's going to increase the stress on the plant.
This is the dead versus wilted leaves. The dead leaves is a pretty direct relation of how fast the plant is dying. This is also an average. You can see there's decimals. We made sure that we didn't measure just the red leaves, and I noticed the other groups had some of those, and we also found those, but we waited till they actually died and turned brown before we measured whether they were dead or not.
So these next two slides show pictures of plants in our experiment. The green stickers indicate the mutated plant, and the yellow stickers indicate the wild type. And during this part, we didn't introduce the saltwater solution yet because we wanted the plants to grow first.
And with this picture, as Berkeley said, the green is still mutated and yellow is wild type. But with the addition of the purple sticker, that's to show which plants get placed in regular water. And so that was our control group, and we placed the purple sticker on it to reduce experimental error and cross-contaminate plants with the wrong type of water. And right after we took this picture, we started applying the saltwater solution.
This is a picture from the end of the experiment. As you can see, a lot of the plants are dying. You can see the bolts and the soliloquies. It's where the reproductive yield, the rosette size would be the distance from one of these leaves to the opposite side. And you just measure the widest one. Dead wilted leaves, you can see a lot of them on this plant, not on this one so much.
So our results did not support our hypothesis because we first hypothesized that the mutated experimental plant would die first. However, the wild type experimental plant died first. As you can see, the wild type plant had the highest number of bolting layer experiments. Also had the biggest rosette size layer experiment, and also is tied with the mutant control in having the most number of soliloquies. So it just shows that the wild type plant reproduced faster in order for it to be able to die first. And this was caused because the AAP2 gene was still activated in the wild type plant, and so amino acids were still carried out. And with that saltwater solution, even more water was still being carried out. In the mutated plants, however, the AAP2 gene was inactivated, and so amino acids and the water was still being contained in the cell.
So the entire purpose of us doing this experiment is to apply this research to the field of agriculture, because as you know, there's plant pathogens out there that reduce crop yield and could potentially cause famine, such as the great potato blight. And so we just wanted to see, what does this gene affect, and see if we can apply it to agriculture safely to increase crop yields and to reduce the risk of famine. And as you've seen with this data that Berkeley has shown us, that the mutated plant is overall increased hardiness, not only to the virus, but to the salt water, which could be a very real problem for countries like Australia that have saltwater intrusion occurring in their country. And so that they could apply this research and this knowledge to other plants, because Arabidopsis thaliana isn't, a cash crop or something that you can really eat. But if you could apply it to corn, say, which is very big in America, I don't know about Australia, but if you can apply it to a plant that's the staple for your country or your population, then you can just increase crop yields and reduce the overall risk of famine and make your country kind of a better place.
Real quick. When you had the two side by side, yeah, right there. So are the two experimental plants both? Okay, never mind. I couldn't see the one line at the bottom. So it's the wild type experimental that has shown the most dead and wilted leaves?
Yeah, along with the mutant
Mutant
control,
control.
the lines.
Okay.
So we label it.
Something maybe to think about, especially those of you in Goochland Block 3, is the very next presentation. It's like we use the two opposite ends of kind of salinity tolerance for these plants. So it's like you guys got the slightly weaker solution, and then Block 1 for orange, we use the stronger solution, and see a difference for sure. So it's just interesting to see how big of a difference that was between them. So you saw, I guess, effects of salinity on the plants when they went to 4 grams, right? 4 grams per liter. You noticed that it, like the experimental plants seem to be okay, I guess, with that. Okay, just wanted to make sure for like I said, I want to compare kind of my own head. So, all right, thank you.
Oh,
Yeah, before we leave the graph, how would you interpret the result from the rosette size? Because, you know, at first glance, it looks like the,
Bring it back.
oh, thank you. The, let's see, the wild type control and the experimental mutant have same diameters.
The only thing we could think about this is we thought maybe they were like over top of each other. Maybe it didn't grow as far out, but with the other data, we thought maybe it was just a smaller plant. Maybe it grew more up instead of out, and so it didn't. It didn't quite fit with what we thought, but with the other data, we thought that the experimental mutant did do better than the other.
Okay, thanks.
All right.
Wow, you guys all know what I'm like.
Okay, guys, we're going to take a break in a couple minutes, but we got a couple more presentations before breakdown, so please give your full attention.
You guys, if you decide to go all on one side, the camera's right here. So when you're standing on this side, you're out of camera. So if you're going to all stand on one side, stand on that side.
Yeah, it's actually the camera's covering both sides of the screen,
Well,
but we get more to the back of your head from this side, so. Just saying. Okay. All right, please welcome the orange, Block 1 group.
Arabidopsis thaliana is known as a model organism for many reasons, some of which include its short life cycle, its close relation to the mustard family, and its small genome that can be easily modified. In this experiment, the AAP2 gene was genetically modified to be missing and not show up in Arabidopsis. The AAP2 gene is a gene that is located in the cell membrane, and it's a protein that transports amino acids and sugars to the rest of the plant. The purpose of this experiment was to show how salinity affected the growth and function of Arabidopsis without the AAP2 gene. Salinity was already known to stunt plant growth. This is because when a plant encounters salinity, it releases a hormone called abscisic acid. This causes a complete halt in root growth. With the less area for the roots to absorb nutrients, it causes it to have stunted growth. It is hypothesized that the mutant plants will have a harder time transporting amino acids. It is already known that the mutant plants have more seed pods. Since salinity is known to stunt plant growth, it should be expected that the experimental plants should have a harder time growing. It is also hypothesized that the mutant experimental plants should grow worse of all because of the double negative in nutrient transport.
For this experiment, there were 2 types of Arabidopsis thaliana being tested, wild type and mutant. The wild types are the plants with the functional AAP2 gene, and the mutants are the plants without. One of each of these types was put in controlled conditions, receiving normal water, and experimental conditions, receiving 8 grams of salt per liter of water, creating the 4 groups of Arabidopsis tested in this experiment. To start the experiment, the wild type and mutant seeds were separated into 2 pods each and planted. Once the plants began growing, they were thinned out by using forceps to pull out the extra sprouts so the 5 plants left would not be overcrowded and have plenty of room to grow. Throughout the experiment, the plants were watered by filling up a plastic container a quarter of the way full of water and placing the control plants inside, allowing them to soak up the water necessary through their roots. The same was done with the experimental plants, except it was filled a quarter of the way with saltwater solution. Throughout the experiment, the plants were also measured in rosette width and bolt height by using a ruler, and the seed pods were counted so we could analyze our results.
As it can be seen in this graph, there was not a significant difference in the quantitative data for the 4 groups of plants. It started about with the same growth rate. In the middle, there was slightly different growth rates for each of the plants, but by the end, they all ended up being about the same. So, what was seen, there really wasn't a size difference. This picture shows that the rosettes are about the same size for each plant. There really weren't any unexpected results because we hypothesized that the wild type control would be the most successful, but the experimental would be least successful. However, because it was such a small difference, we can't 100% say that that is accurate. It could have been due to error that this occurred. So, the one thing that was different, though, we noticed in the 4 groups was coloration. The controls were mostly green. The experimentals had parts where they were yellow because of the salinity.
As it can be seen in graph 2, when comparing the mutant control and wild type control, there's almost no difference in the growth rate. Throughout almost the entire experiment, they had almost the same growth rate. But at day 32, there was a difference. The mutant control had a sudden drop in bolt height growth, where the wild type control was continuing to grow. And in comparing the wild type mutant